In this video, what I'd like to do is go through the process of DNA replication. We now know that Watson and Crick's model was largely intact and accurate, that there's a double helix, and Watson and Crick themselves even predicted that their model was going to be easily explainable in terms of replication. So here we go, a short lesson on DNA replication. Like I said, Watson and Crick did receive their Nobel Prize in the later part of the 1950s for their work with DNA. And one of their quotes was pretty telling at the time, and it said, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copy mechanism for the genetic material." So in short, they basically predicted that there was an easily explainable way to replicate DNA. So 1958, Meselson and Stahl carried out a very important experiment to determine whether DNA replication was conservative or semi-conservative. And when I say conservative, I mean that would require the double-stranded helix to totally replicate itself into two new daughter strands, which would be together, versus semi-conservative, which means the individual strands of the duplex of the parent DNA would be replicated into new daughter DNA to create a hybrid. So that is the big question that emerged. Is DNA replication conservative or semi-conservative? Well, they proposed that the replication was semi-conservative. That means that each daughter strand contains one old strand and one new strand. So here's Meselson and Stahl's experiment. Initially, they took the original DNA and they radioactively labeled that with nitrogen-15. Well, remember that nitrogen is very prevalent in the nitrogenous bases, A, C, T, and G. They applied that DNA to a centrifuge, and because it was radioactively labeled, they could find the density of that DNA. Well, that DNA, not surprisingly, was very dense and formed a band pretty low in that test tube. Because it was bacterial DNA, it replicated really quickly. In fact, 20 minutes for each round of replication. That allowed them to study the first round of replication in short order. After one generation, Meselson and Stahl found that there was no N15 DNA, but rather all the DNA was what they called a hybrid, half N14 and half N15. Well, if you think about it, that's perfectly consistent with the semi-conservative model of DNA replication. If each parent, which was heavy, split first and then replicated with light DNA, then one would expect both new strands to be half N14 and half N15. So it really did make sense regarding DNA being a semi-conservative replicator. To be sure, they allowed it to go for one more round of replication. And the results kind of confirmed what they believed was true. After 2 rounds of replication, the hybrid DNA was there in perfectly equal amounts with the light DNA. Well, that makes sense because each of those hybrids must have split apart before being replicated into 100% light or N14 DNA. So 1/2 of the DNA strands after 2 rounds of replication were light. And we still had half of the DNA being hybridized. So this experiment clearly showed that DNA replication is semi-conservative. And of course, that just demonstrates the bands and how they showed. So N15 initially, we had a hybrid after 1 round, and of course after 2 rounds, we had 2 equal bands, one being the hybrid and one being the light DNA only. So this is kind of what the picture looked like after Meselson and Stahl. we have the original parent DNA somehow must split apart semiconservatively, and each of the new strands is replicated from the parent strand after that duplex splits apart. The question is, how indeed does this DNA split apart without becoming all tangled up like an old slinky and coming back together again to make two new daughter strands? That is the question that we'll look at next. Well, the first step of DNA replication we call initiation. And understandably, the DNA has to unwind to make the A's, C's, and T's and G's available. Replication has to begin at what's called the origin of replication. And we'll talk much more about this. The unwinding is done by enzymes called helicases and topoisomerases, and we call the point of unwinding as the replication bubble. This is where the double helix actually splits apart. Those hydrogen bonds between A and T and G and C have to come apart. Here's a picture that shows this. So remember that DNA is an antiparallel molecule. The top strand in this case runs 5' to 3', and the bottom strand runs 3' to 5'. This is a replication bubble right here, and of course replication would occur technically going in both directions from that replication bubble. This picture just shows many replication bubbles occurring simultaneously, and that makes sense. If you've got 4.5 billion base pairs in the human genome, 1.2 meters of DNA, we can't just count on one replication bubble to do all the work. Many of these must occur simultaneously. The electron micrograph that I pointed out here kind of shows this. You can see the individual replication bubbles occurring simultaneously as the DNA is replicated. This one here on the far right is bigger than the two on the left. Steps 2, 3, and 4 involve elongation, and this is quite a complex process. DNA is replicated, you'll remember, using the parent and is produced from the 5' to the 3' end. The leading strand goes from 5' to 3', and the lagging strand also goes from 5' to 3'. We'll talk in a minute regarding what the leading and lagging strand actually are. So, the steps in DNA replication. This is a good picture because it shows things with quite a bit of detail. Firstly, the helicases involved have to unwind the DNA. So you could imagine that initially, as this DNA was just starting to unwind, as it's moving to the right, those helicases are unwinding the double-stranded into two strands. These are a group of proteins that help achieve this task. So that's the first step. The second step is that RNA primase, which is another enzyme, has to lay down RNA primers, which is interesting to think about, that RNA is actually initially laid down on the DNA template to make new DNA. And that new DNA is synthesized from 5' to 3'. Next, another group of enzymes called DNA Polymerase 3 synthesizes a continuous strand of DNA using the leading strand. And in this case, when we talk about the leading strand, that's called leading because this replication continues to happen towards the replication fork as that DNA is unwound. So leading strand synthesis actually occurs pretty continuously as we move away from the replication fork. Let's look at lagging strand synthesis. It's a little more complex because in this case, replication is moving away from the unraveling of the DNA by helicases or the replication fork. Well, there are similarities between lagging strand synthesis and leading strand synthesis. Number one, primase, once again, has to lay down individual primers of RNA. These are pretty short, and as it lays them down, it kind of allows DNA polymerase to get a foothold and start replicating new DNA from the end of each primer. Those sections of DNA that get laid down in between the primers are known as Okazaki fragments. Interestingly enough, Okazaki was a Japanese scientist who actually ended up dying of radiation poisoning from the bombs that were dropped in Japan at the end of World War II, and he committed himself to learning more about DNA replication. So there's the RNA primer. That allows lagging strand synthesis to occur away from the replication fork. Finally, in order to seal the deal, as we say, with lagging strand synthesis, another form of DNA polymerase called DNA polymerase I has to kick out the RNA primers that were laid down previously by primase, because we can't leave RNA inside of our DNA replication. So that primer gets released, and DNA polymerase, kind of like a bulldozer, pushes that RNA primer out of the way and helps complete the DNA replication on the lagging strand with new DNA. Finally, another enzyme called DNA ligase seals the nicks. The little gaps that occur between the Okazaki fragments, really important because we need a continuous strand of DNA on the lagging strand, just like we had on the leading strand. This series of diagrams really clearly shows both leading strand synthesis and lagging strand synthesis together. Like I said before, leading strand synthesis is very easily achievable, and that's because other than the RNA primer laid down initially, the new DNA is laid down toward the replication fork kind of unimpeded. As long as the helicases are unwinding the DNA as the replication fork moves to the right, leading strand synthesis can occur. Lagging strand synthesis, on the other hand, is more complicated. And like I said before, the Okazaki fragments that get laid down individually have gaps in between them. All right. The primers that are there have to be removed. And of course, DNA polymerase I will help achieve that. And DNA ligase will seal those nicks as we move away from the replication fork. So if you can imagine, lagging strand synthesis has to keep kind of moving away from the fork and backtrack to continue on as we move that DNA unwinding to the right. This picture just shows in more detail DNA ligase coming in to seal the nicks on lagging strand synthesis. And when it does so, we end up with a duplex of DNA with no gaps. Really important. Finally, we get to termination. This is the point where DNA replication has successfully occurred, and we've got two new double helices that are formed. If you think about it, though, the DNA that has been newly formed in some places might be single-stranded. That makes it extremely vulnerable to little enzymes called endonucleases, which I tend to refer to as molecular scissors. And if we've got too much DNA left behind as single-stranded, those molecular scissors will bust it up into 1,000,000 pieces and render our genetic blueprint inactive. That would be catastrophic. So the rewinding of DNA after replication has to happen pretty quickly. And you can see here we've got our new strands laid down. Right here we have our leading strand being produced toward the replication fork and our lagging strands being produced away from. So one would imagine that both leading strand and lagging strand synthesis are occurring at one point while the rewinding is occurring at another. So it's a very dynamic process, to be honest. So we've got one more topic to talk about, and that is the fact that DNA polymerase, like you and I, does make mistakes. In this case, you can see that DNA polymerase has incorrectly tried to lay down an adenine instead of a cytosine that's supposed to combine with this guanine. Luckily for us in our DNA, this DNA polymerase makes a mistake quite rarely, about once every 100,000 base pairs. When one considers, however, that the human genome has 4.5 billion base pairs, an error every 100,000 creates a lot of errors, and that would be unacceptable to the organism. So what have we got? Inside of our DNA, we have an enzyme called DNA polymerase II that works slower and more methodically. And what it does in a very clever way is that it recognizes these incorrect bases, such as this adenine, and it excises or removes it. And instead, it allows the new DNA to be laid down correctly with the DNA polymerase. So instead of having the incorrect adenine laid down, we can now go back and put the cytosine in that should have been there originally. So there you have it. That's DNA replication in a nutshell. Incredibly complicated, and when one considers that it all happens in the small nucleus of the 60 to 100 trillion cells in our bodies, quite an amazing process.